Hi everyone, my name is Alireza. I'm a postdoc in Christina Leslie's lab at Sloan Kettering Cancer Center. And today I'm very happy to talk about our recent work learning multiomic velocities of dynamic germinal center B cells. So the outline of my talk is as following. First, I will talk about the importance of RNA velocity in dynamic systems like germinal center B cells. And then I will introduce our new method called the DynaVelo, which is a joint dynamical modeling of single cells using multiomics data. And then I will show how we can use these models to kind of learn the regulation patterns of RNA and motif velocities. And finally, how we can use these models to kind of do in silico perturbation and find the perturbation targets to kind of restore the loss of function. So I should also mention that this is a collaboration with Ari Melnick's lab at Weill Cornell Medicine, who generated the data. Okay, I want to first briefly talk about the germinal centers. So we had one wonderful talk today talking about this germinal center B cells. These are part of lymph nodes where like antigen activated B cells enter inside these germinal centers, and they have two zones, dark zone and light zone. The cells enter in the dark zone and they undergo proliferation and somatic hypermutation to improve the BCR affinity, and then they migrate to the light zone. In the light zone, for follicular dendritic cells and T follicular helper cells present the antigens. If the BCR affinity is good, these cells get survival signal and they recircle back to the dark zone to undergo another runs of somatic hypermutation and proliferation to further improve the BCR affinity. After several runs, these cells where the BCR affinity is good, they might differentiate towards plasmablast or pre-memory B cells and finally exit the germinal centers. And if the BCR is not good, they might undergo apoptosis and the cells will die. So as you see a lot of dynamics here, a lot of circularity. So our goal is here to learn these dynamics using multiomics data. So how we can learn this dynamics? So we either have to have time or some like notional velocity. Unfortunately, we don't have time. A lot of single cell data is just one snapshot. So that's in our case. So we have to kind of stick with the velocities. So okay, what is RNA velocity? So RNA velocity, based on this SCVelo paper, the assumption is that as following. We have three main events: transcription, splicing, and degradation. And each of these events have their own rates, right? So they assume that these splice and unsplice rates, so they follow two ordinary differential equations. They kind of map this data, they fit this data to this ODE, they learn these parameters, they learn some latent time for each gene, and finally they do some heuristic to map this latent time for each gene to a latent time for each cell. So what we end up here is a partial RNA velocity matrix because we don't have information of all the velocities for all the genes. So the genes for which we have, quality control over a number of spliced and unspliced counts. So, but I will show that we can use our model to kind of recover all of the velocities. I should also mention that we have different samples, wild type samples and samples where we have mutations in ARID1A, which is an epigenetic regulator, and also CTCF. And for ARID1A case, we also have heterozygous mutation or homozygous mutation in one copy or two copies. So, and these mutations are coming from the lymphoma patients. So our collaborators did all this. And ARID1A is part of a canonical BAF complex, which is a chromatin remodeler. They kind of they are recruited by pioneer transcription factors, and they kind of change the landscape of the accessibility. So why we care about the velocities? So sometimes it's very hard to see the changes in RNA expression space. So these are our samples. So some wild types and some of them like ARID1A mutant samples and CTCF. As you can see inside this big block, which is the germinal center, everything is homogeneous. We can't see any differential pattern. If we do any gene expression differential analysis, and we can see clear differences between different genotypes here. But something is happening, because if we look at the attack space, this is just LSI space, we see like we have massive change in the germinal center in the ARID1A mutant samples. They have their own island kind of create, which makes sense because ARID1A is a chromatin remodeler. If we have mutation on this, it's going to change massively the chromatin landscape. So when we have this massive change here, what we can see in the expression space, we wonder why. And our hypothesis is that the velocities are much more sensitive. This is an analogy that I like every time I'm talking about the dynamics. Suppose that we're driving a car in a road, and we have like a positive state, which is the expression, and the derivative of state, which is the velocity, and the derivative of velocity is acceleration. We have all of these positive, we're going with positive acceleration, and then we push the brake pedal, and suddenly the first change we see is acceleration is negative, but we still x and v are positive, and then we stop, and if we go backwards, the velocity starts to be negative, and finally the states become to be negative. As you can see, the order of effects of perturbation is from higher order dynamics to the lower order. So that's why, like looking in velocity space, we can it's much better than expression. So it's much more sensitive. So our hypothesis here is that the epigenome and transcriptome, as you know, they co-regulate each other. So we have cis regulatory elements around genes which kind of control the expression of these genes, and the expression of transcription factors do other downstream changes, and can change the motif accessibility of other. So from attack side, what we're interested is the motif accessibility of these transcription factors. So we assume that, so we have a joint dynamical model instead of just one. So the rate of change in both expression and motif accessibility are kind of jointly a function of each other. And our goal here is to learn these functions, fx and fy, and also a latent time for each cell, because this is also hidden. We don't have any understanding. So we have this full matrix of RNA expression, motif accessibility. We have some partial knowledge about the RNA velocity from SCVelo, which we use to guide the dynamics. And byproduct of this model is motif velocity, which is the rate of change in motif accessibility. We call it motif velocity. So I'm not going to the details of the model. So this is just architecture that we're using, a variational autoencoder architecture, because the observation space could be huge. We can learn this ODEs in that space. So we bring this down to some lower dimension space. And in that lower dimension space, we model this dynamics with neural ODEs. So basically, these functions are very expressive, and they can learn all kinds of dynamics. So we learn this dynamics here in the latent space, and then we map them again to the original space to reconstruct RNA expression, motif accessibility, and also RNA velocity and motif velocity. Here also we're supervising on this velocity, partial velocity information that we have. So we have two vector fields, RNA space and motif space. Cells kind of have joint dynamics in these two different spaces. So, okay, what kind of genes we are using? I don't know why we can see this Venn diagram here. So basically, it's the combination, a union of highly variable genes, velocity genes, and also all transcription factors that are expressed in at least 100 cells, because we're interested to see the effect of regulation of these TFs on the other TFs. So this is what we see. So this is one of the samples, wild type samples. So it's very interesting that we can recover all the dynamics that we care. We have this proliferation and cell cycle inside dark zone. We have transitioning from dark zone to the light zone. We have recircling back of this central sites to the centroblast, and we have these two exits toward pre-memory and plasma cells. So this is a summary diagram of all these dynamics that we care here. So, and this is also showing the cell cycle effect that we can clearly see. So if we look at the latent time learned by this model and compare it with the SC-velo latent time, we see that the cells are following the trajectories of these velocities much more consistent with the velocities than the SC-velo. So we can learn different lineages, so like three main lineages that we see, plasmablast one, the cell cycle one inside the centroblast, and also the transitioning from centroblast toward the centrocytes and exiting towards the pre-memory cells. So we can look at the dynamics of all important and interesting transcription factors based on these four modalities. We have RNA expression, RNA velocity, motif accessibility, and motif velocity. Sometimes it's very hard to see the changes in RNA expression and state, but it's much easier to see in RNA velocity because these are much more sensitive. But the idea is that if the expression is increasing, we should have positive velocity. If it is decreasing by time, we should have negative velocity. And if it is kind of stable, the velocity should be kind of zero, white color here. So yeah, and then here we see that if we, like usually one analysis that people do, they correlate the RNA expression with motif accessibility. And we do the same with RNA velocity and motif velocity, and we show that it's much more sensitive, and we can see better effect sizes. Like, for example, two factors that we're interested is SPY1 and NF-kappa B2. We see for like both of them like positive correlation, but in this other case, we can't see that. So this is a generative model. We can generate the trajectories. Like this is, I'm showing 10 random generated velocities starting from the root cells inside the dark zone. Some of the trajectories are going towards the centrocyte. Some of them recircle back again to the centroblast. So we can look at these generated velocities. We can look at their kind of, this is the trends that we see based on these generated ones. So expression, RNA velocity, motif accessibility, and motif velocity. And we can clearly see that these velocities are really the derivative of the other curves that we see here, which is what we want to see, but it's hard to see in real data because it's very noisy. So these are two important genes that we like to see always because of our collaborators' work. And the idea is that in centrocyte lineage, the first SPY1 motif accessibility goes up, and then later on, NF-kappa B accessibility catches up. So these two are working together to kind of, regulate the germinal center reaction, which they recently published in this cancer cell paper, which is the idea is that in wild type cell, this PU1, which is the SPY1, is pioneer factor comes to bind to the DNA, recruits the BAF complex, and then later on NF-kappa B2 comes, and they kind of control the chromatin accessibility state, which is interesting to validate all of these findings. So after we have these models, we can look at these Jacobians to say something about the gene regulation. And here we can talk about four different modalities, because we have motif accessibility, motif velocity, and RNA velocity. We can see the effect of each modality on another, so effect of RNA expression on RNA velocity, expression on motif velocity, and vice versa. We have all these four Jacobians here. I'm showing just two of them here. So this is just the effect of expression on expression. And we say how the transcription factors, so it's hard to see here, but each row and column is TF here. So when it's positive, it's red. This means that they are working together to upregulate or downregulate their target other TFs. So it's a network. This is just kind of a summary of this network of TFs. It's very hard. So I've summarized the important ones on the right side. So the most important positive and negative regulators, as we see some of the important factors that we care, like FOXP1, SOX5, PU, A1, F1, and PAX5. SPY1. So all of these factors that we care, we can find here, and also the other modalities in a similar way. So we can map this. I don't know why this doesn't show these U maps. So we can add this kind of edges, so this regulation on these U maps to show which ones are differential and different lineages. So finally, we wanted to see if we can validate some of the known germinal center regulation. So from this review paper, which talk about the transcriptional regulation of memory B cell differentiation, these are some known. Of course, this is not perfect, but it's just some well-known transcriptional regulation, kind of a minimal GRN here that we want to see if we can validate some of that. And interestingly, like if you look at the wild type here, some of them we can validate, but like we can do this in cell state specific way, because gene regulation is not static, it's dynamic. So it's changing over different cell types and cell states. So we can show, and we can show also that in ARID1A mutant samples, we lose some of this regulation. For example, the negative regulation of hex on BCL6. So it should be negative that we see here, but it's kind of we're losing that negative regulation in the other ARID1A mutant samples. So another way of looking at this is to look at in silico perturbation instead of Jacobians. Basically, we in silico knock out these factors and we look at the change in velocities, how the RNA velocity is changing and how the motif velocity is changing. So basically, they're similar things, but the effect sizes could be better because this is bigger knockout compared to the Jacobian. So, and then we can map this, like for example, I'm showing here just two regulations, effect of BCL6 on PARC2 and IRF4 on BCL6. So we can see also we have the power to have two kind of GRN. So usually when people talk about GRN, it's an RNA expression state, but we can also talk about the effect on motif accessibility. So two kinds of networks here. And it's interesting that we can see here, for example, on expression regulation, we see the first expression regulation comes, and later on we have regulation on the motif accessibility. So it shows that these could not be synchronous, right? So this could be some cascade event over the latent time. So, and finally, what happens in the mutant samples? So this is one example, very blurry, I don't know why. Basically, the idea is that the centrocytes in the mutant samples can't recycle back to the dark zone to undergo another rounds of somatic hypermutation. And this leads to a very fast exit towards pre-memory. And these pre-memory cells become immature, as our collaborators also validated in their work. So as you see, if we increase the mutation rate in ARID1A, we have expansion in pre-memory. So instead of very slow exits, instead of like letting the B cells to really improve their BCR, they quickly exit without having a good BCR. So this leads to immature pre-memory B cells. And what's going wrong? So these are some differential patterns in the velocity and the motif accessibility. For example, CTCF goes up, right? CTCF goes up, and these two factors that we care, like SPY1 and NF-kappa B2, they go down. So we can see these differential effects here, and how we can predict this. So we do in silico perturbation of ARID1A, and we show this like change in velocity. As you see, this is very messed up compared to the wild type velocity. So this shows that the cells can't have their typical dynamics in the systems, and these are the genes for which we can accurately predict this direction of the effect. So we have mutant versus wild type in experimental setup, and our predicted knockout versus wild type. And very interestingly, the two genes that we see here, SMART-CA2 and ACTB, are also part of BAF complex, like ARID1A. So it's kind of that they are working together. And also we can predict accurately that CTCF goes up and SPY1 or SPY-B motif goes down. So which is very interesting to see that we can validate some of these in silico perturbation. And right now we're working to apply this to the mutant samples to find the perturbation target to restore this effect of ARID1A loss or CTCF loss. So as a summary, we developed DynaVelo and modeled the dynamics of germinal center B cells using both RNA expression and motif accessibility. We learned the RNA velocities, motif velocities, and latent times of the cells. We learned some regulation patterns within and across these two modalities. And finally, we did some we predicted in silico gene perturbation and looked at the effects on RNA and motif accessibility. And the preprint is coming soon. And finally, this is our lab, Christina Leslie. I want to thank her for all her support, and our collaborators, Ari Melnick, and the funding sources. And with that, I'm happy to take any questions.